So say we have 8 modules, and say we only navigate with the smallest one, you're almost always going to end up in the wrong location. If you have a very large module, you're roughly going to hit the goal location, but the signal might be noisy, so you're not going to hit it precisely. So the idea is you start looking at the largest module, and then you go closer, and then eventually it's going to yield responsibility, and this module is going to take over, this module is going to take over, and then eventually, as you get closer, you also get higher precision, higher resolution signal, and you're going to accurately hit the goal. So that's the idea of how you can navigate with grid cells. Now we get to the distortions. So you see here that the pattern might compress if you compress the box, but only in one axis. So now you suddenly have it's an elongated pattern, and we have the shearing. So what happens in navigation model with distorted grid cells? Well, let's first look at just one grid module and say it's distorted in this way. So now the grid pattern is skewed like this. What we see in the brain is that all the cells within the module are distorted in the same way. So going back to this neural sheet we talked about earlier, you're still going to have a bump of activity in that sheet. It's just going to shift around differently as a function of how we actually travel in the real world. But this means we can still try to just use the same decoder as before. So with no changes to the decoder, the result looks like this. So we still have a region where you're able to navigate. It's just that the trajectories might be a bit curved. But other than that, this looks fine. And then, say we then have 8 modules, and we've distorted them in different ways. So this is shared, this is shared, this is shared a different way, and then this is compressed like this is compressed like that. But the main point is that one grid module only needs to bring you one step closer, so the next module can take over. So if we look at the regions where one given module is able to navigate, and we look at these ellipses here show where the larger scale module will deliver the agent, these ellipses are all contained within the regions of the modules. So we see that we can navigate with one module, and we see that the handoff from a larger module to the smaller module should still work. So looking at this, it looks like navigation might still be possible. And indeed, we can navigate, even though the modules are distorted, and they're distorted differently. So that's the simulation results then showing that with this particular setup for navigating with grid cells, distorted grid cells shouldn't be a problem. But what does this then mean biologically? So it's useful to then consider why does the nested grid cell decoding work? And the main feature of this model is that when you're far away from the goal and you're mostly navigating based on the large scale modules, you're not really looking at the smaller scale modules. So when you're far away from the goal here, you're mostly navigating based on the signal from this grid module. You don't really care what this one is doing, so it might be completely jumbled up, and you're still going to be able to get closer. And then we can sort of assume that as you get closer to the goal, this module might be able to perform error correction. They recognize a familiar landmark or something. So I don't think this is implausible that if you travel far away, you have noisy path integration, it's going to drift. So it might look like this, but you get closer and then it's corrected. So nested grid cell decoding is quite robust to these noisy conditions we might expect biologically. And the point is just, one module needs to deliver you to the next, and that's it, that navigation will work. So in this situation also, with those just completely jumbled grid cells, you can still navigate. The thing is, with nested grid cell decoding, we're making a quite strong assumption that you have a large number of grid modules. And we don't really know if this is true biologically. So, biologically, 4 or 5 grid modules have been observed concurrently from one animal, but then they haven't really examined the whole part of that cortex simultaneously. So this is still an open question biologically. Are we limited to on the order of 4 or 5 grid modules, or might we have, say, 20, which is a more reasonable number if you want to navigate with nested modules? So we see that with nested decoding, if you can assume that you have the large number of modules, then these problems of distorted modules and noisy path integration, for example, shouldn't really be a problem. But if it turns out that we only have a few number of modules, then the situation is that we can't use nested decoding, and then another proposal is that you might do something called a combinatorial decoding, where, say, this is the largest scale module you have, that module starts repeating, and then you might still attempt to navigate beyond the range of this largest scale module because the conjunction of activity pattern across all modules are still unique because they don't repeat at the same frequency. So, but then it's a question of how do you do combinatorial decoding, and combinatorial decoding seems less able to handle grid distortions because they actually depend on these tightly interlocked conjunctions of activity across all modules. So, yeah, we don't yet know how the brain uses grid cells. Are grid cells used for vector navigation? Is that then combinatorial, nested, or something else? So that's an open question, but at least nested grid cell decoding, it's a cheap way of decoding grid cells. We saw we only needed 12, 13 or something neurons to decode that module. And the approach is quite simple, and it can handle distorted grid cells, perturbed grids, from noisy path integration and so on. So this should be a quite viable thing to put in a robot, for example. And then, is this biologically possible? We don't know yet. So neuroscientists need to look more in the brain to see how many modules there are, for example. And then in the meantime, we can keep building these models and making theories, evaluating them. And I think this really shows also the potential benefit from looking at the brain, trying to build something, and then trying to go back to the biological system. So thanks for listening.
Thank you, perfect timing. So we have 5 minutes for questions from the audience. So do we have questions? Yes.
You are probably familiar with the study by McGuire about the London taxi drivers. And what he found out is that basically this population of taxi drivers, they have a larger hippocampus. And so there's one thing that strikes me. In your model, it seems that we can have overlap of representations by using this combinatorial decoding. You can have overlap that is somehow hierarchically you're going to represent any place you've ever visited, right? It's not possible to have a grid cell for every place, but if you have modules, you can represent all that hierarchically. So if you think about these London taxi drivers, do you think they have more grid cells or more modules? What would be your idea here, your hypothesis?
Yeah, so my immediate thought is that I have to memorize different unique locations, and so they need to memorize different places, different routes, and so that's more in the place cell system, whereas I think the grid cell system is more general. It's for doing computation on two-dimensional vectors, computing differences, calculating angles, and so forth. And so whether or not you have very good knowledge of the street layout of London, the length of the vectors a long distance runner or something like that would traverse longer distances in absolute space. Whereas I think the taxi drivers would need to they would need to memorize more stuff about different locations, so that would more be place cells, which are adjacent to grid cells in the brain.
They need to know the fastest route to go from one place to another, so this is a navigation task for them.
Yeah, and can grid cells participate in route calculations like that, or is that more a topological problem of how the places connect to each other? Maybe these vectors you can extract from grid cells aren't that useful, but that they would be, for example, more helpful in large open fields in a huge forest or something where you don't have that many obstacles as you would in a city environment. Thank you.
Thank you very much.
Other questions?
So there is a recent study by DeepMind, which is the training the network to do a path integration leads to a grid cell leads to the emergence of grid cells. So if your research is to do with such one research, please explain the relationship between the
Yeah, so my understanding of that work is that they arrive at similar capabilities in the agent, but through machine learning. So I didn't get into all the technical details here, but all of the stuff in my model is handcrafted. So and the grid cells aren't even neurons; they're just explicitly generated to generate those grid patterns. But and then the decoding part is also handcrafted. And what they show is that you can do machine learning to arrive at some similar capabilities. You get the grid cells, and then you also get the vector navigation capabilities. So I think it complements this work nicely. Yeah.
Do you think the underlying mechanism is similar to the result from the machine learning, or is it completely different?
Yeah, so in the paper I don't really see any discussion of how the network actually performs the vector navigation part, how it does the decoding. So I think that would be interesting to see what do they actually arrive at through the machine learning.
Thank you.
Yeah.
So I think we have time for one last question while the next speaker prepares. So I also have questions actually. So what does it mean that the grid cell is distorted? It's not the cell that is distorted, it's the sensory field, I guess. And is that learned or it's hardwired?
Yeah, I think the mechanisms behind these distortions is still an open question biologically also. So in the model it's just hard coded into the model, but why does this happen and why does it develop over time? It seems to be affected by sensory inputs, but yeah, it's still an open question.
Okay, thank you very much.
Yes, thanks.
So the next speaker is Jason Yoder, who will talk about behavioral stability in the face of neuromodulation in brain-body environment systems. So, you're ready? Okay, good.
Okay. Hi everyone, I'm Jason Yoder. I'm a PhD student at Indiana University studying computer science and cognitive science, and I'm going to talk about a project worked on the past year with Ecuardo, Izquerido. There. So, brief outline. I'm going to introduce the concept of neuromodulation briefly, why it's important, why you should care about it, and look at a particular paradox in biology that's an open question. I'm going to talk about a model that we're using to explore this task, go into some results, some analysis of understanding a system, and then relate those back to biology at the end. So, neuromodulators are chemicals that are released in the brain. You've certainly heard of them before: adrenaline, serotonin, dopamine. And they are differentiated from neurotransmitters in that they generally affect a region of tissue, and they're not simply excitatory or inhibitory. They can have other types of effects. One way of understanding them is functionally rewiring a network in a different way. They also have been tied in with enabling or disabling learning. So you have often a diffusing chemical cloud that can then impact neural activity within a region of the brain. So why is this important? Well, if we look at even the simplest model organisms that we have a full connectome for, like C. elegans, 302 neurons, we know exactly how they're wired together. You would think if we had all that information, we know where everything's tied to each other, we'd be able to predict behavior and neural activity. Well, as many of you know, that's not the case. We really can't predict things, even for such a simple organism. And neuromodulators are a very significant part of this. If we don't know what neuromodulators are present in the system, and we don't know what receptors for those are present, then we really can't predict these behaviors. One way of understanding this is that there's an abundance of connections, there's over-connections, and by introducing neuromodulation, we can selectively choose what circuits need to be activated at any given time. So, the big question right now in biology regarding neuromodulation is this. How can we have a network that's so sensitive to changes in concentrations of even a single neuromodulator can totally alter the way that it works, while at the same time having the stability that organisms are routinely and robustly performing particular behaviors, despite the fact that these things are changing throughout the lifetime. And there's dozens of these in living systems. Just a diagram to see a little bit of how complex they can be. So trying to understand this, there's been some work done, but it's usually done in isolated circuits in a very limited way. And to try to tackle this question, I want to try to answer what types of mechanisms could allow this behavioral stability to be maintained. So Eve Marder is a very prominent researcher on neuromodulation, and she had some suggested mechanisms. She had a proposal of 5 of those. And these are largely proposals, ideas, but they haven't been evidenced in models one way or the other. And so I was hoping in my project to demonstrate perhaps some stability mechanisms. And the nice thing about the whole ALIFE approach and using computational models is that we can have an entire system and we can show plausibility. We can show that, in principle, if we have certain assumptions about our model, that we can show that things will work in a particular way. So with the model, what we'd like to do is show that, first of all, we have a model of neuromodulation which can disrupt a system. Then we want to show that we can maintain the same behaviors even as a potentially disruptive signal is coming in. And then finally, if we can identify what enables stability, being able to perform behaviors even as something disruptive is coming into play. So, used a model that's been built before. It's been analyzed very thoroughly. It's a relatively simple model, but that makes it very easy to go and analyze. So the original idea for this task was insect locomotion, having a very simple model of legs, just walking with sticks. Analysis of this showed that you can understand the generating dynamics of locomotion from just a single leg. And so that becomes a nice model. If we have just a single leg that we're being controlled by a few neurons, we can try to understand exactly what's going on. And the nice thing with the computer model is that we understand everything going on in the neurons and body all at the same time. So this is a visualization of the idea of the model here. We have 3 motor neurons. There's one neuron that's responsible for lowering or raising the foot. And in the picture here, video here, you can see when the foot is down, it is red. So it goes down, it pushes back, and it makes progress across. There's a neuron that controls a backswing motion and a neuron that controls a forward swing motion. So those oppose each other. But if it's successfully coordinating these 3, it can make progress stepping, and it can only push the body forward if it has the foot down on the ground. Now, there's a couple different models that we use, a couple different models of these walkers. One type is going to include this angle sensor. So it's going to have some feedback, some proprioceptive feedback that's telling effectively this walker where is the leg in relation to the body. So it has a little bit of sensory feedback. That's one that we varied, though, a bit. So the one that hasn't is a reflexive pattern generator, or RPG, I'll refer to it. So that's the one that always has that input. There's also central pattern generators, which have no input. They're totally driven internally, so they don't have any kind of feedback from the outside. And then there's another version, mixed pattern generator, which sometimes has that feedback and other times does not. And so those are 3 different types that we're using here. And the model itself we're using here, again, based on prior work, continuous time recurrent neural networks, and then the neurons are also receiving input not only from each other, but if they have that sensory feedback, it comes from the angle sensor, and they get one signal when it's at one end of the body, and the opposite from the other end. So, all of this up to this point has been the model that we chose to look to add neuromodulation to. So, reproducing work, something that could be done more often, I think. This is a case of actually reproducing past work. So we did develop and get all these different models to work and walk proficiently. Throughout the presentation, we have tested the mixed pattern generators when they don't include the signal and when they do include the signal from the leg angle sensor. So you can see some differentiation there. But the main point here is that all of these different walkers can evolve to be proficient at their walking behavior. No big surprises. So now what we'd like to do is we'd like to see, okay, can we disrupt these with a neuromodulatory signal? So the form of neuromodulation we use here, don't have a ton of time, but I'm going to just point out three main things about it. First of all is that it's extrinsic. It's not controlled by the network. It's something that's influencing it. Think of it as hormones released by the body as opposed to something that's released directly within the network. It is not doing a regular excitatory inhibitory act. What it's doing is it's effectively multiplying the signals, either amplifying them or dampening them. You can think of that as the signals themselves or the weights of the connections within the network. It's unavoidable in that all neurons are influenced by it equally. And when we're testing the network, we give them enough time to take 50 steps if they were a perfect walker. So there's an idealized perfect walker that could take 50 steps in this amount of time. And then we vary, are we amplifying or dampening the input throughout the entire time? So right here, each of these steps, it effectively is getting tested at a variety of different levels. So we wanted to have a measure for how well does a walker actually cope with this signal. So what we did, rather than just having a single test, we decided to vary the amplitude, so having different degrees of it, not just this is a maximum level, 50% greater, 150% or 50% less. Just do a series of different tests and see how much of the time are they performing at least 90% of the best performance. So, this work, this is the same plot from before. Over here we have, you'll see that the central pattern generators were very disrupted by this. Really all of them were substantially disrupted by it. The reflexive pattern generators, some of them did pretty well, but overall we have a pretty severe disruption of performance. So, what we'd like to do is we'd also like to have results showing that we can have behavioral stability despite this very disruptive signal. So, to do this is relatively simple. If we just put the neuromodulation present during evolution, that will allow for the potential to select for stability mechanisms. And because we're doing an evolutionary search, we're taking out, for the most part, human bias in the process. So, when we do this, we can see that up here we have similar high-performing walkers. So even in the presence of neuromodulation, they can still evolve to walk quite well. And it should be relatively unsurprising that then when we test them with varying degrees of it, whenever there's less of a signal, it does not influence them nearly as much. So this in and of itself is not too impressive, but if we go and we try to understand what's going on here, now maybe we have a chance to gain some insight into mechanisms. So we want to know what's the difference between the networks that can walk well and the ones that don't. And can we connect any of those mechanisms to things that have already been proposed or maybe some new ones? So to do that, we're going to start by looking at one particular network, and then we'll see how neuromodulation affects it. So again, this is actually a video of this particular network. If we think about what's going on during this walking motion, and we look at the readouts of the different neurons, we'll see that whenever the foot's down and it's pushing back, we have basically a fixed state for the backswing neuron. As it's pushing back, it's fully on. So right up here, we have that foot pushing down, and the backswing is on. And then once it gets to the tail end of the body, it stops pushing back, it transitions, and goes into a state where now we have the foot. The foot is going to be off, and our forward swing is going to move it back to the front of the body. So this is a repeating pattern happening over and over again, and so this can be understood as 4 states, call them quasi-stable states, because we have several neurons that aren't changing state and others that are. So 1, 2, 3, 4. The first one, we have a thick line to indicate that they're on, so the backswing and the foot are on. The gray indicates that it's changing from being on to off or off to on. And then we can see the connections between. Okay? So this is effectively a way we can understand the dynamics of what's happening. So we'd like to test and see what point does this network get disrupted. So this network is definitely breakable by the neuromodulation effects, and so at what point does it break and why? So one thing that can help us a little bit to understand what's going on is a steady state input-output plot. So this is a way of visualizing what's going on to a particular neuron. So if we're looking at the backswing neuron, if I inject a constant, very strong negative signal to it, it's going to drive it down, and it's going to be sitting there just a stable point off. Similarly, if I put a very positive signal, it's going to drive it upwards, and it's going to reach an equilibrium point at the top. However, because a neuron has a self loop, there's a potential, if it has a strong self loop, that depending on the starting state, it may go down or up based on a certain amount of input. So if we're at 0, if it starts at a low enough state, it'll go down. If it starts at a high enough, it'll go above. So this is useful to understand and visualize what's going on within the network. If we think of what happens as it's transitioning between these states, it starts where it's on, comes off. As it changes, though, the thing you have to remember is it's not just this one. As this one goes from being on to off, it's affecting the other 2 neurons that are in the network. And so then the amount of current that comes into it will change. So instead of being fixed at that point, once it changes state, now it's going to transition over, and so it has this same cyclic pattern as before. So whenever we apply neuromodulation, we decided to apply it at fixed levels to see what is the breaking point. At what point will the walker stop moving? So in the dampening form, where we're reducing the amount of signal coming in and reducing the weights, it got stuck at this particular state here, where the neuron's trying to go from off to on. So normally it goes up, but it hits an equilibrium point, and the only factors that are influencing it here is an excitatory connection from the forward swing neuron, and because we have a negative bias, so that's one of the parameters for CTR-NNs, is going to be keeping it down. As we dampen that signal that's coming in, it's going to end up not being able to overcome it, and it's going to reach a fixed point here, and it's not able to move. So that's one thing. If we go to the opposite end, if we go and we look at the amplifying modulation, what ends up happening here is normally we have the neuron go from being in a positive and on state, high firing rate, and then going down. And what's driving it to do that transition is a inhibitory connection from the foot neuron. So that particular connection has a strength of -3, but the neuron itself also has a self loop of +15. Now, there's a difference between those by quite a bit. So if you amplify the signals to the entire network, now that neuron is going to have a strong tendency to want to stay on, and the normal inhibitory connection, which is amplified as well, isn't amplified as much. And so, again, we hit a fixed equilibrium point. Okay. So if we selected from the neurons that the set of networks that we evolved to walk with neuromodulation present during evolution, that were very proficient and were able to walk stably in the presence of it, we find 2 different mechanisms that enable this stability and this behavior to continue. So in the normal state where we ran into trouble because of the dampening modulation, which we weren't able to escape, weren't able to go up, right? We got stuck on our way up. In this particular walker, we have a positive bias instead of a negative one. Now, despite the fact that it has a different bias, it still had that same cyclical pattern of which neurons were turning off and on in the exact same pattern. But because it had a different transition mechanism, because it had just a positive bias, that allowed it to go up in spite of that. So that's one mechanism. The second mechanism, this is a more interesting one, ends up being connected to the biology a bit. In this case, we have the same type of connections, right? We have an inhibitory connection from the foot and a self-loop on the BS neuron, the backswing neuron. But this time, the value on the inhibitory connection is -15, which very closely matches the positive value of the self-loop. So we have two things. They're both being amplified. Because the ratio is very similar, it's going to be able to maintain that behavior. Okay. So that's the central pattern generators. The reflexive pattern generators, and a big part of the story, so I won't talk about this for a couple minutes yet, we found that they were much more robust in general. And in fact, when we look specifically, even at the networks that were evolved without neuromodulation present, they were able to be quite stable at walking if we look at particular values of the modulation signal. So looking at and keeping modulation at an exact level, we can look and see how well the networks did. So starting at this end, dampening, trying to drive down all of the activation, and here amplifying all of it, you'll see here that we have actually a relatively high value of walking fitness for the reflexive pattern generators, and it's dropping down drastically for the other ones. So how is it possible that we can have this stability even though the neuromodulation is present, even though the networks weren't evolved with it present? How can they still be stable? And if we look at this particular slice right here, where we have a maximum amplifying, we'll see that the fitness value has a very high end on the distribution. So walking almost perfectly despite having it present, even though they weren't exposed to it. So to understand that, you have to realize a little bit about the mechanisms of the reflexive pattern generators. So they have these weights from the angle sensor, which will drive them to transition. So the backswing neuron, for instance, if we have a very strong signal, when it gets to one position, right at the front of the body, then it's going to transition it to turn on and to drive back. And so by looking at a distribution of the parameters, we can see that, by the most part, these transitions are being driven from the sensor angle position. So the thing about that is that even if we have a very strong amplifying signal, that's not going to make much of a difference because we still have to wait for the leg to get to the positive end before it turns on. If it's at a neutral position, if you're multiplying 0 by 150%, it's still 0. So the effect doesn't really kick in until we get to the higher end. Run a little short on time, so I'll skip over another mechanism there, but largely the results of connecting this back to biology are pretty exciting. First of all, that first mechanism of the two different connections that had a similar weight, that can be understood as something that Eve Marder talks about as coordinately acting on opposing processes. So if modulation occurs the same on two, it will balance things out. Having a signal that once it's already close to maximum, we try to amplify it and it still stays maximum. We also see evidence of that, especially in the RPGs. The third one, tentatively, that could be tied in with increasing network size to increase performance. But the most exciting aspect of what's going on with this particular result is that we've shown that with proprioceptive feedback, even whenever a network is evolved without neuromodulation, it can potentially still have a relatively robust performance in the face of it. So that's very exciting, and that's something that we couldn't accomplish with an isolated circuit, and gives some exciting news for us using computational approaches that we can explore things that biology might not be able to otherwise. Make sure we have some time for questions, so I'll just leave it there.
Thank you. So do we have some questions?
So do I understand that you are always modulating the entire network?
Yes, so that was an intentional decision to try to make it extra challenging. In biological systems, there's a wide range of different neurons and receptivity. But it was trying to make the task intentionally challenging and intentionally disruptive by forcing all of them to be receptive. There's some evidence of other mechanisms dealing with selective sensitivity to combinations of neuromodulators, but for this particular work, we wanted to try to find something new that hadn't been evidenced.
Yeah, because I would think if you're modulating parts of the network at a time, it'd be kind of like dropout, right? Where you're like making it robust to changing in local environments or something like that. But also,
right.
Is that, I'm not totally familiar with, is that basically how it works in the brain? I mean, is it more regional neuromodulation or is it more total brain?
Right, so there's different forms of it. There are some more local transmissions to a particular area. If you look, there's like dopamine, it gets dumped into particular regions. But there's also volume transmission where it's just going over a large space. It's generally not the entire brain, though. It's usually a particular section. Again, for the purposes of the model, wanted to make it intentionally disruptive. If you let them all just not listen to neuromodulation, it's a trivial task. So, yeah, it's.
How much do you think your neuromodulation could be just modeled by a change in the synaptic efficacy or the weights between your different neurons due to sort of this wave pattern going over the entire network and the limited number of weights? And if that's the case, maybe presumptuously, how much is the neuromodulation experiments that you did just testing in the evolution sort of a good initial state to start a network in that it can then learn from or change from for this specific type of neuromodulation?
Excellent question. So in fact, with the model that we're using, it really is effectively just changing synaptic weights. It's directly the same, so you can think of it as moving within a region within the parameter space. And so that's a good way of thinking about it. Can we find a region that's stable enough to despite the perturbations? So you're spot on with that. That's very accurate. In terms of how realistic is that in the biology, there's tremendous complexity. Very little is known, which is why we took the computational approach. But using a multiplicative effect is one of the common forms. So, right.
Other questions? So I have a question. Here, neuromodulation is a purely disruptive inference, right? And I kind of thought that neuromodulation had something to do with learning or some positive aspects.
Absolutely. So actually, most of the work that I've been doing up until this was trying to look at how to harness the positive effects of neuromodulation, which there's, if any of you are familiar with the dopamine signaling, it tends to tie in with reinforcement learning. There's some good evidence of that. And there's lots of positive ways that it can be used. I've been surprised by how challenging it has been to find them. For some of the tasks that I'm doing, there's sometimes that it is disruptive. And part of this project is to understand this question in biology. If we can understand maybe biology is dealing with the disruptive effects in this way, maybe if we have more embodied models, we can harness the positive effects, but also mitigate the disruptive effects. So that's another aspect of it.
If I can ask another one. In relation to sort of selling this back to biology or talking to biologists about this, a lot of neuromodulation papers I've seen try to exactly mimic a specific neuromodulator. And so have you thought about, or with the inspiration of this sinusoidal sort of global signal, are you going to say this is sort of serotonin or this is sort of histamine?
So a great question. So part of the thing, right? You can try to pick a specific organism because the particular neuromodulator depends on the receptors, what effect it's going to have. But our idea with this was a slow changing, so maybe like a hormone that's changing over time with the day and night, something like that, where you have a slow change, but you need to deal with it and adjust through it throughout the day. And in terms of this, thinking of it increasing the sensitivity, you could also think of it on a shorter time scale, something like adrenaline, where you're more reactive. So I think there's a good argument for a couple ways. Haven't gone and made the very specific ones, but I think there's some good cases for it.
Okay, thank you. So our next speaker, please get ready. So Katarzyna Kozdon, who will talk about the evolution of training parameters for spiking neural networks with Hebbian learning. Okay, so we tested this and it's worked.
Just tested and it worked.
Is the stage manager here?
Oh no.
I guess I'm supposed to fill in with announcements, but I don't have any.
Thank you. Sorry about that. So today I'm going to talk to you about my PhD research in spiking neural network, and today in particular about how using evolution and unsupervised learning in spiking neural network together gives particularly good results. The name spiking neural networks comes from the activation function they used, which is inspired by electrophysiological recordings in biological neurons. So as you can see in the panel on the left, neurons accumulate input, and when threshold is reached, they fire. We use leaky integrate and fire neuronal model, which means that when the threshold is not reached, some of the charge leaks out. However, some of it still stays in the neuron, forming a kind of memory of the previous iterations. Unlike most traditional artificial neural networks, it's very common in spiking neural networks to use bio-inspired spike timing-dependent plasticity to update the weights. And according to this technique, weight update is basically a function of spike timing of two neighboring neurons. We use 3 weight update rules. The first one is the most commonly known one, long-term potentiation, also known as fire together, wire together rule. According to this rule, if the first neuron shown here in red fires first, and then its target shown here in blue fires second within a certain time window, the connection between them strengthens. Another popular rule that makes the weight decrease is when the firing pattern is reversed, so the target neuron fires first, and then the first neuron, the red neuron, fires second. Then the weight decreases, and the rationale behind it is that the red neuron was not necessary for activation of its target in the first place. Additionally, we use a neural that was inspired by findings in the brain. So in the brain, as probably most of you know, we have two main groups of neurons: excitatory neurons, which increase the potential of their targets, and inhibitory neurons, which decrease it. It has been reported that if the first neuron, the red one, is inhibitory, the firing order doesn't matter. As long as the two neurons fire together within a certain time window, the connection between them strengthens. So there are some problems when it comes to spiking neural networks. As I said, they don't use backpropagation, they don't minimize any error. This means that learning in them is not directed. They do not know what you try to do with them. And their performance is worse than traditional artificial neural networks. They are still not popular or good enough for real-world applications. Similarly to the first speaker today, I'm somewhere at the intersection of neuroscience and AI. I take biological algorithms, try to understand them, and see if we can apply them to AI to improve it in some way. So last year we published a paper on parallel processing using ensembles of those spiky neural networks to take advantage of what we see in the brain a lot, parallel processing of data. Today I'm going to talk about evolving learning hyperparameters for those spiky neural networks. And when it comes to design of our experiments, we use inputs which are inspired by biological experiments in animals such as rats and monkeys. During those experiments, you have an animal facing a screen, and on the screen it can see moving shapes, high contrast shapes, and brain activity of the animal is being recorded. So similarly, we design 4 simple black and white shapes, which are placed in the visual field at a random location. And then they consistently move in one of the permitted directions, left, right, up, down, with the speed of 1 pixel per iteration. And the way those bitmaps are mapped onto the neural network is by vectorizing them, and then mapping 1 pixel per 1 input neuron, in such a way that white pixels do not affect the state of the neuron, whereas black pixels always affect the neuron fire. So the input is mapped onto the network. During training, as the network fires, the weights are being updated. During testing phase, we analyze the activity of the output neurons. We do it by creating a binary vector which indicates spike timing of each individual output neuron. So here we have an example with 4 time points, and here the first output neuron fires at time point 0 and 3, and does not fire at time point 1 and 2. So we end up with 1 binary vector per each input we show to the network. And then we use self-organizing maps to cluster them. The rationale behind it is that if the spiking activity of the network is very characteristic for the type of the input it was presented, then self-organizing map should clearly separate vectors coming from different input classes. Then we score the results and basically calculate precision. During training, we show each shape for 5 iterations, and in total we show 20 shapes moving in one of the 4 directions per training epoch. During testing, we show each shape for 100 frames, and we show all possible combinations of shapes and directions. So in our previous work, we used even a simpler input. We used just vertical and horizontal stripes. Still, the networks do not perform as well as we would like them to, because as I said, they do not know what you try to do with them. In order to be able to work with more complex patterns and to improve our results, we decided to try evolutionary approach. And according to this approach, we are trying to develop, to optimize 4 different learning hyperparameters for our spiky neural network. The first 3 hyperparameters are related to the weight update rules I told you about, long-term potentiation, long-term depression, and inhibitory long-term potentiation. And basically those hyperparameters specify the size of weight update when those rules are applied. The 4th hyperparameter we're evolving is discharge, which defines how much the target neuron, how much its state is updated. And discharge is identical for all neurons in the network and it is multiplied by the weight for each pair of the neurons. So here, networks can inherit two different things: the learning hyperparameters we are evolving, or weights, meaning experience of their parents. And inheriting learning hyperparameters without any training is meaningless. We start by initializing a population of 30 spiky neural networks. We train them, we test them, and we select 10 best networks. And we propagate them according to 3 rules. We either copy them directly, just as they are. So this allows us to test the well-performing networks on another data set to see if they are good at generalizing. According to the second rule, we copy them, but then we take them through additional round of training to see how this affects performance of the network. And the third rule is we copy the best 3 networks, we introduce a mutation in one of the learning hyperparameters, and then we perform training so that learning hyperparameters can affect the weights. So we started by trying to optimize the networks to recognize movement direction in the input. It doesn't matter which shape they are being shown or where in the visual field the shape is. We just want them to cluster the results based on the direction of the movement. And in panel A, you can see fitness of the population, performance of the best, 3 best, and worst network. And as you can see, even in generation 1, we're getting very good results. The precision is nearly optimal. And I'm going to get back to this in a few slides. When it comes to performance of the worst network, its performance is similar to just random guess, 1 in 4, but as you can see, it improves with time. In panel B, you can see the number of optimal solutions in the population, meaning number of networks which achieve 100% precision. And as you can see, this number increases in population as evolution progresses. Here, each color indicates a separate experiment, and this is to show you that initially, evolution explores the whole permitted range of hyperparameter values, but then, with time, it converges on a narrower range. We also compared weights of the parents and children with either mutation and training, or with training alone. And without going into details, the 2 main observations were that the number of weights that are being updated decreases as evolution progresses. One of possible explanations for this is that some of the connections become silent. Second observation is that later mutations cause larger bidirectional changes in weights. So as I said, in the first baseline experiment, even in the first generations, we were getting results that are close to optimal. So the question is, can our approach generate optimal solutions and not only propagate them when they are already present in the population? To test this, we worked with a smaller population, and we selected only networks which were performing below 95% precision. So here, the average performance of the best networks in the first generation was 88%. However, in the second and third generation, in all experiments, the optimal solution appeared in the population. And as you can see in panel B, the number of optimal solutions in the population starts to increase with time. This suggests that this approach can generate optimal solutions when they are not present in the population. Next thing we wanted to understand is the role of evolution versus training in the improvement of this performance. We cannot completely separate evolution and training because training is necessary for the evolved hyperparameters to actually affect the weights. So first we looked at fitness in the absence of training. So this is basically survival of the fittest. We repeatedly train networks and choose those that are best at generalizing. And as you can see, I appreciate the difference between the baseline, the first experiment, and this is not big. But when we started those experiments, before we used evolution, we were not able to get any meaningful results with those networks. But as you can see, the performance is below 100%, and in panel B, the number of optimal solutions in the population does not increase with time, and some of the optimal solutions are being lost during evolution. This suggests that evolution alone is not enough to drive this improvement in accuracy. We also looked at the role of weights and how inheriting experience of the parents affect the performance of the networks. In panel A, you can see detailed fitness of weights which are being evolved without inheriting their parent weights. They inherit only the evolved hyperparameters. In panel B, we compare those networks which inherit just the learning hyperparameters and networks which inherit both the hyperparameters and experience. And you can see there is no significant difference. This suggests that when you evolved promising good learning hyperparameters, the experience of the network does not matter. It can still learn. We also looked at random parent selection. What was the role of survival of the fetus in improvement of the performance? And again, you can see that the performance is slightly lower than the baseline. And in panel B, the number of optimal solutions is relatively stable throughout evolution. So those last three slides suggest that it's the combination of evolution and training together that drive this increase in precision. So all previous experiments were focusing on clustering inputs based on movement direction. Next, we wanted to look at clustering results based on shape. And we expected this to be more difficult because spiking neural networks, because of this internal memory inside each individual neuron, are meant to be particularly good at processing time series data. And as expected, results in this experiment were lower, but again we see improvement of precision as evolution progresses. And even the worst network that starts with random precision improves and reaches about double precision, starting from 25% precision to about 50% throughout the course of evolution. In panel B, we look at the values of evolved learning hyperparameters when we're optimizing the networks either to detect movement direction in gray or shape in black. And two hyperparameters we were evolving showed different preference depending on what we were optimizing the networks for. So discharge and long-term depression. And in case of discharge, when we're optimizing networks to cluster the inputs based on shape, the evolution preferred low values of discharge, which means the network was far more quieter and that spiking pattern was far more subtle. So hopefully I convince you that using evolution and unsupervised learning in spiking neural networks together is a promising approach, that we achieved faster convergence and better stability of fit solutions in the population, as well as higher fitness of overall population. And seeing as I still have some time left, I wanted to say that spiking neural networks in comparison to traditional artificial neural networks are still quite small fields. So thank you for letting me to give you a talk about this. And I already met some people who work in this field here, and it was a great pleasure to talk with you. And I'm hoping to see more research happening in spiking neural networks in the future. Thank you for listening.
Thank you very much. So do we have some questions?
Sorry if you're tired of me. Fantastic work, really, and especially your note about spiking neural networks. Not too many people use spike time-dependent plasticity also. What was really surprising to me about this work is that the constants for the inhibitory spike time-dependent plasticity seem to be on par with the long-term potentiation. Considering the sort of novelty of inhibitory spike time-dependent plasticity, and its reduced use seemingly in biology, could I ask about your sort of inhibitory excitatory ratios in the brain and the connections there, and which weights you were training with which parts of spike time-dependent plasticity?
So ratio of excitatory inhibitory neurons.
Mm-hmm.
If I remember correctly, it was either 15 or 20% of neurons were inhibitory, which is kind of similar to the number reported in the cortex. And connections are feed-forward, and it's random decision which neurons are excitatory and which are inhibitory.
All right, and then the excitatory weights were, all of the weights were trained by all three different types of spectrum-dependent plasticity that you had?
So if the presynaptic neuron was inhibitory, it was using the inhibitory update rule. But other combinations were using the two first rules, LTT and LTP-like rules.
All right, thank you.
Do we have other questions? So I also work on spiking networks, so I have a million questions. But so one question is the fact that you obtained very good performance from the first generation. Could it mean that your parameter space is so small that random initialization is enough to get good results?
It seems like it, but when we tried, even with simpler inputs, to initialize them by hand, we're consistently failing. And especially that we're working with an ensemble of the networks and we wanted to get a nice set of networks, it was impossible to do it by hand. So there's a chance that some of it is just the advantage of working with a population of the whole generation. But then, as we can see, when we start testing the effect of evolution and training, having them together further improves it, so it's more than just random search.
Thank you. So I have another question, if nobody has questions. So maybe you know that there is a lot of controversy about the shape of the STDP function, and you evolved LTP and LTD, right?
Yes.
So did you observe any interesting shapes or?
I didn't look at the shapes. Taking account that I'm not exactly modeling the firing rates, I suspect that the exact shape of the update might be different than what is observed in biological neurons.
But what is observed in, so many things are observed in biological neurons. Like some people say STP is like this, and some say like this. So even if you find something that is a little bit different from what you expect, it could still be interesting.
Mm-hmm. I will look into it, and I'm sure we will talk after.
Any more questions? Okay, thank you. So this session is finished. I think after this we have the poster session. So in one room you have the posters. And in the other room, we have the late-breaking papers as posters. And I think before that, we have a coffee break.
Do you have mine? Do you have mine?